Hello and welcome to week 3 of Biology 1124's lab. Before we get into the lab this week, I just want to go over some housekeeping things. So I've gotten a couple of emails about procedures for each week and clarity in what the assignments are, and so I just wanted to give a quick overview of the procedure that you'll be going through each week. It'll be similar, and if there's anything different on a given week, I'll send you an email so that you know ahead of time. So the first thing you should do every week is read the lab background. There are a couple questions in each lab background. They're not exceedingly difficult, but they will make sure that you know the things that you need to know before going into each week. While we're not collecting them or double checking that you do them, so it's not required that you do them, it will help you. And I just really want to emphasize that even if we're not collecting it, it's a good idea to do them ahead of time. The next thing you should do is watch the intro video that I'll be posting each week Monday around noon. You're already watching one, so presumably you already know about it, but just to go over it again. After you watch the intro video where I'll go over the procedures that you'll be watching each week, you'll watch the experimental video, and I think it's really important that not only do you record the data that you're observing, but you should also be taking notes. What color changes are you seeing? What procedures are going on that maybe you want to ask for clarity. Just don't write down the data and skip through everything else, because watching it is going to be important so that you understand what's going on. After that, you'll be writing your lab summary and sort of synthesizing all of the information that you just got from the video. These should be 1 page long. Don't stress too much about the word limit. Try and stay around 300, but if you go a little bit over, that's okay. A little bit over. I don't want to read like 500 word, 2-page things each week. And then after you do that, you'll be uploading a video to Flipgrid. Each week you'll need a new link to that week's assignment, and I'll be sending those out as well as including it in the description of the video that you're currently watching. So this week, we're going to be going over a colorimetry lab. Virtually, you'll be carrying out an experiment and collecting data. And the point of this lab is to look at enzyme activity and the different conditions under which enzymes are doing their job. Afterwards, you'll be writing your lab summary and uploading it to Canvas, as well as doing the Flipgrid video, which I just went over. So in lecture, we've been talking about enzymes and their activities and how they work under different conditions. And last week we talked about and learned how they are proteins, and they're strings of amino acids, and they're really important for actually carrying out a lot of biological functions. And so this week we're going to be looking at how enzyme, in this week's case catecholase, how it operates under different environmental conditions and which ones are the best for that enzyme. So today's intro video, we're going to be going over a lot of the equipment that you'll be using and the procedure that you'll be following, or that you would be following if you were in the lab. And so this does seem a little silly to be going over it virtually, but in future labs you may be using some of this equipment, and it's important to understand how the equipment is working and how the procedure goes if you're not going to be carrying it out yourself. But this equipment, or this week, the equipment you'll be using is this colorimeter, which will be measuring the amount of light that is able to pass through a solution. You'll have the cuvettes, which is just the fancy term for the tubes that go into the colorimeter. And then you'll be having the tubes and the droppers and everything that you need to actually transfer these solutions. So the potato cell solution will be prepared in advance, and so you don't necessarily need to worry about how it's prepared. You just need to know that this potato cell solution has the actual enzymes that were living in a potato. So this is an actual live enzyme experiment. These aren't manufactured or anything. So this is just extracted from the potatoes, and the instructions for the lab protocol and the walkthrough and everything should be on Canvas. So enzymes require typically pretty specific environmental conditions. So how much salt they need in the environment, the pH, and of course, temperature. And so all of these are going to affect the shape of the enzyme. And so as you guys may know, the shape of the enzyme is pretty much the entire way that it works. The enzymes need to be a particular shape to fit in with the substrate, which is just the molecule that it's binding to. They fit together like puzzle pieces. And so if you change the shape, you're going to be changing the function. And so in this case, the way that we're changing the temperature is through the ice water baths and the hot water baths. And you'll be using tap water as the room temperature bath just to make sure that your conditions are similar across your treatments. As always, you need to be cautious when using these. The water is hot. It's a hot water bath. So you need to keep it covered. With these things, I can tell you from experience, the steam condenses on the top, and then when you go to pull it off, all the hot water tries to run down on your arm, and you don't want that. So when you're using these things in the future, be very careful when you are taking the covers off. Organization is going to be very important. The tubes that you see in the lab video will all be labeled with masking tape. If you write it on the vial, it can get wiped off, so it's generally better to use tape. But in this case, you'll need to be making sure that all of your treatments are separated. So here you see the pH 3 tube is labeled. You'll be having like the pH 7 and the pH 10 tubes. And yeah, just keep them organized. At this point in the video, you would have all of your tubes and your treatments set up, and the last thing that you would do is add the catechol. And so this is where you have to kind of reason your way through what's going on in the experiment. In each of the tubes, there's a solution made from potatoes, and so in that solution, there's already going to be catecholase. You already have the enzyme there, the thing that's doing and carrying out the reaction. But if it doesn't have any substrate or any catechol, in this case, to break down, you're not going to detect a difference in the catecholase's activity. So you have to add more of the substrate to the tubes, and you want to make sure that you're adding an equal amount to each tube. So then, like most lab machines or whatever you're using to measure anything, you're going to have to calibrate the colorimeter, and typically this will be done with a negative control or a blank where it's just water. So you'll read through how to calibrate it. When you do this in the future, you'll have to refresh your memory on how it works, but for this week's lab, just know that the colorimeter was calibrated ahead of time. When you're ready, you'll be transferring some of your sample to a cuvette, and remember that's just the fancy term for the rectangular prism that you're using to put into the colorimeter. It's a specific shape, and the colorimeter knows how transparent the edges are, so you have to use the specific tool that comes with the colorimeter. And so with these, there's two clear sides that are on opposite ends and then two fuzzy sides. When you load it, you have to make sure that the two clear sides are actually what's being measured, the light going through it. So this is where, again, you need to really think through what's happening. You're going to record the percent transmittance. It'll be a number between 0 and 100. And the transmittance is the amount of light that is able to get through the solution. And so we know that catecholase breaks down catechol into a darker substance. And so when you're recording these transmittance, think about if catecholase is functioning well, would we expect the solution to allow more or less light through it? So if catecholase is going about its job and breaking down catechol into this darker substance, what would you expect the transmittance to be by the end of it? So as I mentioned last week, or 2 weeks ago, I guess, you need to very thoroughly clean your tubes and cuvettes after each test. In this case, it's more important. Well, it's the same amount of important, but if you don't clean your tubes in between your own experiments, there could be carryover enzyme or carryover catechol, and it might be different between the tubes. So you need to make sure that you're cleaning everything out very effectively while you're doing this. When you've finished, you have to rinse everything with DI water, which is deionized water. It's just very clean water, which all of the labs, or a lot of the labs at OU, have an actual tap that will dispense the DI water. So then you just leave your equipment clean and organized for the next lab. To be successful this week, again, you should read the lab background and follow along while watching the experimental video. The lab background is going to have all of the information you need to actually understand what's going on in the lab, and then more importantly, link what you're learning in lab to what you're learning in lecture, and so that you can do well in those lecture exams. So you're also going to want to review the how to write a lab summary document. And definitely look at the assignment rubric before you write, and then look at it again after you write, and then look at it again before you submit. The rubric is how I'm going to be grading these, so just try and even just check it off like a list. Make sure you have everything there that needs to be there. And then I would say probably most importantly, if you have any sort of uncertainty or question or anything like that, come to office hours. Office hours are going to be a really important tool for you guys, because it's a lot easier to convey your question and get an answer from me when we're talking through it, rather than putting it in an email and then waiting for my response and then, you know, trying to get back into the mental groove of things once I respond. So I would say, you know, come to office hours with a list of questions that you may have and get them answered, and then you'll do well each week. Especially on these lab summaries. So they're going to be due at noon on Friday. I know that last time there was a little bit of confusion because Canvas said midnight, and then I said it was at noon. In general, they're going to be due at noon unless there's another mistake. So just make sure that you get it in by then. Your Flipgrid post is going to be due at the same time, noon on Fridays. I'll be including the link in the description of the YouTube video and an email that I'll be sending out each Monday. You'll need a different link each week so that you get to the assignment for that week for my sections. So just keep an eye out for that link. In each video, you'll need your name, a brief summary of the experiment, and the conclusions. Also try and include your hypotheses, because that's the important part of the conclusions, is was what you expected to happen what actually happened. You'll also need to include a challenging part of the activity and something new that you learned. Going through some of these videos, I noticed a lot of students forget the something you learned, so just make sure you put a post-it note reminding you to say that at the end. So that's all I have for you this week. And again, if you have any questions, feel free to shoot me an email or just show up to office hours. Again, those are Monday, Tuesday, and Friday mornings, 8 a.m. to noon. And so hopefully I'll see you there.